Hello, everyone, and welcome to today's graph reading group.
Where Osama tells us about his paper PepFlow, where he develops a Boltzmann generator for small peptides.
Thanks everyone for coming, and thanks for the invitation to discuss my work. So I'm a PhD student at the University of Toronto, supervised by Dr. Philip Kim, and I'll be providing a brief overview of our recent preprints on basically trying to develop an approach that would allow for the direct sampling of conformational ensembles of any input peptide sequence directly. So peptides are, as a class of molecule, are widespread across biological processes, but they've also been continuing to garner increased interest as therapeutics. Currently, there are over 170 peptides in clinical development, and peptides do compose a substantial share of the global pharmaceutical market. In terms of the...
If you say there's 170 peptides in clinical development, do you know how much stuff there is in general in clinical development?
So I don't really have that figure in terms of like the fraction, but yeah, so it is still quite a large number to be in the clinic.
Okay. Anyone in the chat knows, then write it in the chat. But let's see. Lower cost of, yeah, let's go.
Yeah, so peptides sort of occupy a special niche in the range of therapeutic molecules, somewhere between small molecules and larger biologics like antibodies. Compared to small molecules, they tend to have higher specificity and present a lower risk for toxicity. And compared to larger biologics, they tend to have a much lower cost of production, and they exhibit better tissue penetration and cell permeability. So as I'm sure you all know, in the last few years, there's been substantial advancements towards the single state structure prediction of biomolecules, with the foremost example being the development of AlphaFold for the prediction of structures for globular proteins. So this success has to some extent translated to peptides, but nonetheless, attaining a comprehensive understanding of the activity and function of biomolecules is contingent on being able to understand the full range of conformations they can assume. And this is especially true for peptides because they tend to be highly flexible. So as a simple example, the binding of a peptide to a protein is dependent on its ability to assume conformations that allow it to interact with the protein and its propensity to fold into these conformations. So the goal of this project was to sort of move past single structure prediction and develop an approach that would allow for the direct sampling of conformations from the energy landscape of any input peptide sequence. So conventional approaches for conformational sampling that are rooted in statistical mechanics are typically trajectory-based. So these approaches rely on the definition of physics-based functions over the atomic coordinates of a particular molecular system. And given the energy of a particular molecular system, you can then, up to a normalization constant, compute the probability of that particular state at equilibrium based on the Boltzmann distribution. So to actually sample from these distributions, these conventional trajectory-based approaches would start from some region in the energy landscape and take small steps based on atomic forces. Now, this process is extremely computationally expensive, and these methods are susceptible to getting stuck in local minima. And this is further exacerbated by the fact that even for peptides, certain transitions and folding patterns occur over long molecular timescales. For example, beta hairpin formation can take up to 10 microseconds to occur, and that type of time, simulating that type of time scale can take on the order of days depending on the hardware being used. So one prominent paradigm in using deep learning for conformational sampling, and the paradigm on which the work in this paper is based, is the Boltzmann generator. So Boltzmann generator attempts to rectify the issues with trajectory-based sampling by instead using a normalizing flow to directly sample from the target Boltzmann distribution. So the idea is a normalizing flow would be used to map from a simple base distribution to molecular conformations for the target system. So the use of normalizing flows enables exact likelihood computation for the generated samples, and this in turn enables the two training modes that are used for Boltzmann generator. The first being training by example, where the model is trained to match the distribution presented in the a training data set of known conformations. And second is training by energy, where the model is trained to try and match a Boltzmann distribution parameterized by some physics-based molecular force field. So to avoid the invertibility restriction that's present in canonical normalizing flows, PepFlow is trained as a continuous normalizing flow that instead learns the dynamics of an ordinary differential equation that can be numerically solved to go from your base distribution to the proposal distribution and vice versa. Now, I'm not going to go over diffusion models, as I'm sure everyone here is intimately familiar with them at this point. But one key finding from the work done on diffusion models is that for continuous time diffusion models parameterized by a stochastic differential equation, there exists an equivalent flow parameterized by an ordinary differential equation that has the same marginal probability distribution at each time step. Based on this, PepFlow was initially trained as an SDE using denoising score matching, which is substantially more efficient than doing maximum likelihood training with a continuous normalizing flow, and then subsequently used as a normalizing flow for conformational sampling and for training by energy. In fact, PepFlow was actually trained in multiple different phases to try and rectify the scarcity of available peptide structural data. Initially, PepFlow was pre-trained on protein fragments from the protein data bank, and this data set constituted roughly 40 million data points, and was subsequently fine-tuned on bona fide peptide structures. This data set was around 4,000 data points before being further fine-tuned on molecular dynamic simulations and performing training by energy. Now, in terms of the architecture, the PepFlow architecture is actually quite straightforward. The dynamics networks are composed of EG&N layers that operate on a graph representation of the all-atom peptide structure. So in the graph representation, the nodes represent the atoms in the structure, and there's a series of rototranslationally invariant features that are included in the nodes and the edges. So the main difficulty in developing PepFlow came from the fact that we want to efficiently be able to sample all atom peptide conformations without any a priori knowledge of the generated typology, and while still modeling the interdependencies between atoms that might be far away from each other in primary amino acid sequences. So to accomplish this, we introduced a number of provisions into the PepFlow model. So the sort of main provision that was introduced was that instead of generating all the atoms in the peptide conformation in one step, the model was actually divided into 3 different processes. The first process would generate the backbone atoms, as well as a coarse grain representation of the side chain, being the side chain centroid position. A second flow would be used to generate the positions of the heavy atoms in the side chain, and a third flow would be used to generate the positions of the protons in the side chain. Now, the first flow makes use of a fully connected graph, but the other 2 models use a more sparsified graph based on the interatom distances that were found in the initial generation step. So for the monomer generation,
the generation of the hydrogens in the last step, did you do that because you want the hydrogens, or did you do that just because the energy functions need hydrogens?
So it's essentially because energy functions need hydrogens.
Okay.
There is some notion to using idealized coordinates for hydrogens, and instead of trying to model the true Boltzmann distribution, model the conditional distribution based on idealized hydrogen positions. But I do think it's somewhat theoretically satisfying to model all the positions of the atoms, and I guess from a very nitpicky perspective, if you marginalize out the positions of the hydrogens, that wouldn't be equal to the minimum the lowest energy conformation for the hydrogens. So I do think there is some merit to modeling the protons as well. But yeah, the hydrogens do compose a substantial portion of the atoms in the molecular representation, and we avoid having to deal with like very expensive fully connected graphs by simply just using edges between atoms within 4 angstroms when computing the hydrogen positions.
Okay, and do you train this all end to end?
So for the training by example phase, all 3 models were trained independently, but they were trained together in the training by energy phase.
I see, that makes sense.
So the second feature of PepFlow is that a hypernetwork is used to generate sequence-specific parameters for the backbone dynamics function. So instead of providing the peptide sequence to a conditional dynamics function that's then used to generate peptide coordinates, the peptide sequence is instead provided to a hypernetwork, which predicts parameters that define a sequence-specific dynamics function, which can then be used to generate peptide conformations for that sequence. So high-level idea is that there's necessarily going to be a limit to the complexity and the expressivity of a dynamics function while still allowing for efficient peptide conformation generation. So we decided to sort of relegate some of the model expressivity into a network that only needs to be run once during generation. So hypernetwork can within reason, can be arbitrarily large without really adding much running time to the generation process and can drastically increase the expressivity of the network. In practice, the hypernetwork is simply takes in the peptide sequence and consists of some attention layers that eventually generate a high dimensional embedding of the peptide sequence, which is projected to the weight and bias matrices that parameterize the EGNN layers. So beyond altering the model to allow for more efficient generation, there's also a notion of including an inductive bias within the actual process being modeled to make it easier for the model to learn peptide positions. So instead of noising and denoising absolute coordinates of atoms, we instead noise and denoise relative coordinates of atoms. So each atom is, the position of each atom is encoded relative to its N-terminal neighbor, with centroids and encoded relative to the T alpha and oxygen atoms encoded relative to their bound carbon. And these relative vectors are noised and denoised. So effectively what this does is instead of generating uncorrelated point clouds of atoms, it generates ideal chains when the peptide is fully noised. And this much more closely represents a real peptide conformation. And this makes it easier for the model to generate a real peptide from this noise representation. It is important to note that the EGNN layers still operate on absolute coordinates, so they can still reason about the relative positions of atoms in three-dimensional space. So one final
so can we speak more about that? So you're saying you still have the advantage of being able to reason in three-dimensional space, but the things that you're predicting are still 3D coordinates, right? And then you're taking the 3D coordinates, and from those you calculate changes to the relative position.
Exactly. So the model outputs a change in the absolute coordinates, and those are converted to a change in the relative coordinates. I mean, because the mapping from the absolute coordinates to the relative coordinates is, I mean, it's bijective up to, a translation constant, but since the EGNNs are translationally equivariant, that doesn't matter.
Do you see any disadvantage of doing that instead of doing the absolute coordinate prediction?
So for peptides, it just like empirically, it just improved performance to move to this noising scheme. I can see for, larger proteins where you have complex tertiary interactions over longer ranges, this type of inductive bias may actually make it more difficult for the model to learn because you'll end up with these long changes where the model may have more difficulty folding things back in. But considering that for the most part, peptides are relatively linear and you don't have these complex tertiary interactions, I think this inductive bias is beneficial overall.
What do you mean with like why, when I have a very long chain, why would it then be hard to make updates that satisfy a certain or go to a certain tertiary structure?
I mean, it's strictly based on the idea that you'll have to move the ends of the peptide longer distances to assume that fold. Basically, the rate of diffusion of the ends of the peptide relative to the C terminus relative to the N terminus, like the C terminus of the peptide is diffusing at a much higher rate than the N terminus, and folding that back in would be quite an expensive process. Of course, this isn't really problematic for peptides, considering the relatively short chains, and you don't have these complex folds, but that could be a problem for larger proteins.
So it's kind of it's kind of weird, right? Because one side of your protein, like the C terminus, is not moving at all, and your N terminus is moving very, very fast.
Yeah, in terms of like the denoising scheme.
Yeah.
Yeah, and that could potentially be a disadvantage, but I think just having you know, bonded atoms locally or more close to each other in three-dimensional space in the noised peptide makes it easier for the model to learn the real conformation. So there's a couple of questions in the chat.
Yeah, so the first one, how much computational overhead does this change of coordinates incur?
So I don't think there's really any change in there isn't much computational overhead. I mean, mapping from the relative coordinates to the absolute coordinates is, you know, relatively trivial and is a pretty quick operation.
It should be much faster than any single linear layer added to your network. Yeah. Would this relative noise limit the exploration space?
I'm not really sure I understand the question. Do you mind expanding a little bit?
Yo, then maybe Francesco can speak up at some point, and in the meantime, we keep it going.
Sure. Okay, so one final technical consideration is that the process that generates the heavy atoms for the rotamers needs to be modified to actually allow for tractable likelihood computation over the real peptide atoms. So in the setup I described so far, we're able to compute the joint likelihood over the backbone centroid coordinates, and we're able to compute the likelihood of the rotamer coordinates conditioned on the backbone and the centroid coordinates. However, what we're actually interested in is the likelihood over the real atomic coordinates and not the centroids. So in this naive setup, we'd have to marginalize out the centroids. So to avoid having to do this, the model enforces the condition that the backbone and the joint probability of the backbone and rotamer coordinates is equal to the joint probability over the backbone, rotamer, and centroid coordinates. And this is simply done by having a process that maintains the centroid for the heavy atoms during both noising and denoising. And this is a simple generalization of center of mass preserving flows and processes that have been previously used in other work for to generate translationally equivariant networks. So going back to the chat. So question is, you want to sample this state space of the conformation. Is this limited by the noise type?
We don't really want to sample all the goal is not to sample all the possible state space, right? We don't want to sample everything in R to the n times 3, like where n is the number of atoms. We don't want every possible combination of coordinates. We just do this with, like, if we have complete absolutes or Gaussian noise added to our absolute coordinates until it looks like a Gaussian cloud. We just do this such that we have some stationary distribution that we can sample from, or such that it goes to some stationary distribution that we can sample from. And if we have a better stationary distribution that we can sample from, that looks much more similar and builds in prior knowledge of how looks much more similar to the final molecule and builds in prior knowledge of how a molecule looks like for example in eigenfold, if you're familiar with that, then this is a much better stationary distribution to start from, because the denoising process then has to do less. And here we also have this. We have the information included that our molecule in the end should be a chain at the cost of other things, but this is maybe a better station.
So I guess the question of whether or not, you know, having an ideal chain as the stationary distribution would bias towards a certain type of conformation, I actually think that's a possibility.
Yes, it is, but that's what we want, right? We want bias towards.
No, I mean, but like potentially, you know, causing the model to predict, you know, elongated coils more than, you know, hairpins, even though they might occur with a similar frequency. I do think if the model is expressive enough, though, it should be able to, you know, capture the distribution irrespective of, the biases in the stationary distribution. I think ultimately the stationary distribution is close enough to what you see in real peptides. Even, I mean, it's nowhere near, like, it's definitely not a perfect approximation, but I think it's definitely close enough that, it permits the model to learn these varying conformations.
Yeah, very nice.
I don't seem to. Okay. So I guess to summarize, how the PepFlow approach works is a peptide sequence is provided to a hypernetwork, which consists of attention blocks that generate a high-dimensional peptide embedding, which is then projected to generate the weight and bias matrices for EG and N layers that define the dynamics of a flow or process to generate backbone coordinates and positions of sidechain centroids. These positions are then used to define edges in a graph that's processed by another network to generate, the dynamics for the purpose of sidechain rotamer generation. And finally, a similar process is used to generate the positions of the protons in the peptide conformation. Just checking chat real quickly. Okay. So initially, after we trained this model, we wanted to evaluate whether or not PepFlow could at least generate realistic peptide conformations. So I just provided a set of peptide structures that PepFlow generates to show that, at least qualitatively, they resemble real peptides, and PepFlow can generate varying secondary structures, including helices and strands. More quantitatively, we looked at the distributions of various molecular features, internal coordinates in the backbone, and we found that PepFlow can accurately capture, bond lengths in the backbone, bond angles of the backbone, the torsional angles in the backbone, and can also capture the distributions that you'd see for centroid positions relative to the backbone. We also looked at the ability of the rotamer model to
Wait a second.
Yep.
So to be clear about the setup here, right? You train on your 40 million sequences by example, then you train on 4,000 peptide data set by example, and then you train on your 100 whatever data set by example. That's still right?
So, okay, so to be slightly more accurate, PepFlow is initially trained on the fragments and by example, then fine-tuned on the peptide sequence by example. And then during training by energy, the conformations from the molecular dynamics are used as an auxiliary loss because what was found in the initial Boltzmann generator work, and we also found that here, is if you only use the training by energy objective, the model ends up just preferentially generating conformations from one metastable state. So you'd end up just generating all helices for a particular peptide. So by providing an auxiliary loss using real molecular dynamics data, we're able to get the model to learn something that more closely resembles the energy landscape.
You sort of balance the mode seeking behavior of the reverse KL loss, so training by energy, with the mode covering behavior of the forward KL loss by still training by example a little bit.
Exactly. That's exactly it.
But now you're like, what is your MD data exactly again that you train on?
So
by energy and by example. So
the MD data is derived from a data set of antimicrobial peptides, that were just run in MD and explicit solvent for 400 nanoseconds each. So we just took non-redundant examples from that data set and use that as our training examples. In terms of the objective for training by energy, I simply took sequences from the fragment data set, and we sampled conformations from those, and computed the energies using a molecular force field and trained using that. I mean, theoretically, we could have done random sequences, but we decided to use fragment sequences on the basis that we know those peptides can at least in some circumstances fold into semi-structured conformations, which would make them slightly more relevant than just a random peptide sequence.
That makes a lot of sense to me. Okay, Then let's continue.
So we similarly looked at the ability of the rotamer model to reconstruct amino acid side chains. We found that the rotamer model was generally able to do so quite well, with generally very low RMSDs to the real rotamer side chains. As expected, the RMSD scales with the number of atoms and the number of rotatable bonds in the side chains, with arginine being one of the highest in terms of generated RMSD. To evaluate if the model was generating conformations correctly, even when they didn't match the ground truth, we also looked at a distribution of chi-angles in the side chain, and we found that they generally matched quite well, with the only real exception being cases where we had ambiguous labeling of atoms. So in the case of glutamate, the two oxygens at the ends are identical chemically, and since the model is permutation invariant to these type of ambiguous atoms, we get like a mirrored distribution. But this is more of a feature, I guess, rather than a bug, since this is what this is the behavior you would expect. So we next wanted to assess whether or not PepFlow is competitive with single structure prediction approaches. Of course, and to do this, we evaluated the ability of PepFlow to capture conformations of peptides found in the protein data bank. Of course, PepFlow is a conformational sampling approach, so we need some kind of heuristic to select single conformations from the sampled peptides. So the heuristic we ended up using is simply to cluster generated conformations at a threshold of 1.5 angstroms and to select the centroid of the most populated cluster as a prediction. And on the validation set, this seemed to correlate relatively well to the RMSD to the ground truth. So we compared the performance of PepFlow to AlphaFold and ESMFold, and overall it's very comparable. You could make an argument that PepFlow is slightly better, but the performance difference isn't drastic. Here are a couple of examples where PepFlow outperforms AlphaFold and ESMFold. In the first case, it captures a solid feature of the helix where it's not completely helical at the end, even though AlphaFold and ESMFold predict that. In the second case, it's a coiled peptide that PepFlow accurately predicts in the loop, whereas both AlphaFold and ESMFold prefer the elongated conformation.
So for this, do you try to make sure that none of the peptides were in the training data of any of the models?
Yeah, so this test set is recent peptides. So when we were constructing the training data set, it was, structures before a certain date. And we also did like a redundancy check based on sequence identity.
Okay, and what if we're dealing with large proteins that are hard to predict, for example, for AlphaFold, then what are usual C alpha RMSDs that we expect there? Because here everything is below 1 angstrom. Oh,
so here, so I think the metrics for globular proteins and peptides are not necessarily comparable. So these aren't, these are C alpha RMSDs per residue, because the issue is when you end up with really small peptides, you end up with really low RMSDs, irrespective of how different the conformation is. So here we divide the RMSDs by the number of residues in the peptide. So for, you take the reported value here and you multiply it by the number of residues in the peptide.
And is it maybe a little bit meaningless to look at the C alpha RMSD if we're talking about these rather small peptides where we're then maybe as long as AlphaFold puts it into a helix, the C alpha RMSD is basically 0.2. And we would much more care about the side chains. Is that all right? So yeah.
I think there is some merit to that. But in these, unbound conformations in solution, the side chains are going to be quite flexible. So I think it's hard to benchmark on the side chains. I do think subtle changes like this may not necessarily be relevant when doing docking. Of course, larger conformational changes forming into a loop is more significant. But there's definitely an argument that this benchmark isn't really that significant. I do think it is sort of a sanity check to indicate that PepFlow can at least be used to match the performance of these single structure prediction approaches, and it's generally predicting reasonable conformations for different peptides. But I think what we ultimately do care about, and I'll get to later, is the performance on conformational ensembles.
Yeah.
So I guess this is another point, is that in the test data set, the majority of peptides are actually bound to globular proteins, and this context is missing when performing the single structure prediction. So the single structure prediction is necessarily going to be lacking. So we want to assess whether or not, as we generate more conformations, PepFlow does eventually generate reasonable conformations for peptides. And as a baseline, we compare it to this ModPep approach, which is a conformational ensemble generation approach based on fragment assembly of known bound peptides in the protein data bank. So it's not a deep learning approach or anything, but we do find that PepFlow does at least outperform ModPep in terms of bound peptide conformation generation. So we sort of discussed this previously, but when moving to training by energy, the goal was ultimately to minimize the forward KL divergence between the prior distribution and the distribution generated by transforming samples using the reverse time ODE. You can reduce this loss function by removing terms that don't depend on the parameters, and we end up getting is the expectation over the energy of the generated conformations and the change in log density, which can at high level be thought of as, you know, enthalpy and entropy terms in trying to maximize the free energy of the, or minimize the free energy of the system. So we found that training by energy substantially improved the ability of PepFlow to generate conformations that match the MD.
I'm not sure if this was now so transparent, like how we calculate the likelihood and then match it up to our the likelihood that we get from our energy function, right? So you kind of have here on the left in your expectation is can I consider the U the ground truth?
Yeah, so U is the, you know, the energy computed using the molecular force field, and which is one component of this negative log likelihood.
Yeah, and like U corresponds to the negative log probability, right? Because we have our Boltzmann distribution where we have e to the minus U giving us the probability or the unnormalized probability. And now if we take U, that would be the same as taking minus log of the probability. Yeah. And then we have here so minus log of the probability in here. And then we want to match that up. We want our model to produce the same probability for it as this, and this then to be, and then we take a minus between those two, or the difference, and that difference we want to be 0. And this now is the likelihood of our model, because we're taking the Jacobians of every single time step, right? Like for diffusion model. So every single step that our diffusion model makes, we take the Jacobian of it, and then the trace. Why the trace? Why will give us the likelihood?
So this is sort of the, you know, instantaneous change in log probability from the initial work on continuous normalizing flows. So here it's treated as a flow, so it's deterministic. But this is, you know, it's essentially the term that accounts for the change in the log density when you move from the proposal distribution to the base distribution. This is actually quite expensive. Like this training process is substantially slower than, you know, the training by example using generalizing sort matching.
So why is this not the determinant of the Jacobian?
Yeah, I don't like. We'd have to go through a lot of math to derive this, but this is equivalent in the, you know, continuous realization of this type of framework.
So this is like an estimator, right?
I mean, like here we'd use Hutchinson's estimator to compute the trace of the Jacobian, because computing the trace is intractable, or it's at least very expensive.
Okay, so you don't even compute the Jacobian and then take the trace, instead you use?
Yeah, we use Hutchins' estimator, so
Okay.
you can do that efficiently using a vector Jacobian product, which allows for efficient training. I mean,
Do
with
you
the number of degrees of freedom present in a peptide, this would, yeah, it would just, it's prohibitively expensive. In torsional diffusion, in the torsional diffusion paper, the trace was computed exactly since it was small molecules and only the torsional angles were being modeled. But since all the degrees of freedom were being modeled and it's a slightly larger system, yeah, exact computation of the trace is intractable.
And do you know the details? Okay, is it quickly explainable how the Hutchinson trace estimator works?
I mean, I don't know the proof off the top of my head. How
Not the proof, just how it's done.
Yeah, so you sample from a distribution with mean 0, variance 1, and I can try to annotate. So you sample, epsilon from, we can just say a Gaussian.
Normal.
0, 1, and the estimate would be E times the Jacobian of F. Or here, this would be the transpose. And the expected value of this is equal to the trace.
So we still have to compute the full Jacobian.
Well, you compute it as a Jacobian vector product, which is more efficient in like an autodiff framework.
Okay.
Yeah, which substantially increases the efficiency of computing this loss function.
Yep, very nice, very nice.
Okay, so yeah, in terms of the effects of training by energy, we see higher concordance with molecular dynamics simulations after performing the training by energy. We see a higher coverage of PepFlow conformations by molecular dynamics conformations, and we see a higher coverage of molecular dynamics conformations by PepFlow conformations. We also performed this latent temperature scaling, where instead of sampling from the unit Gaussian in latent space, we instead decrease the standard deviation by applying a temperature scaling factor, and we found that also improved the ability of PepFlow to generate realistic ensembles. So ultimately, what we wanted to do was validate the ability of PepFlow to capture ensembles that were generated experimentally. There aren't that many available peptide conformational ensemble, but there does exist this protein ensemble data set, which contains conformational ensembles derived from experimental data. And this contains a large number of disordered proteins, which tend to be enriched in short linear motifs, which are regions of proteins that independently bind other proteins and generally exhibit peptidic features. So to get an evaluation set, we use the Elm server to annotate short linear motifs in these proteins, and we extracted the various derived short linear motifs. And we found that the annotated short linear motifs had varying degrees of tertiary contacts. So some were located in highly structured regions, like this fragment that's found in the middle of a beta sheet, and others were found in disordered regions and were highly coiled. As expected, we found that the performance of PepFlow is highly correlated to the number of tertiary contacts. So we just used a distance cutoff based on the position of the Cartesian distance in three-dimensional space between residues and the relative position of different residues in primary sequence. So as you can see, the scale divergence of the radius of gyration distributions and the pairwise C alpha distributions increases with the number of tertiary contacts, and likewise the coverage of PepFlow conformations and ground truth conformations decreases with the number of tertiary contacts. So we used a relatively lenient threshold of 3 tertiary contacts per residue to define our test data set. And here's sort of the performance results for PepFlow on that test data set. In general, the radius of gyration generated using PepFlow is highly correlated with the ground truth radius of gyration, showing that PepFlow in general is able to capture whether or not peptides tend to be more elongated or folded. We also find that on average, you see roughly 40 to 50% of PepFlow conformations are captured by the experimental ensemble, but this value is quite highly correlated with the number of experimental models you get and tends to converge at around 1,000 to 10,000 conformations. So when you have an extensive experimental models that capture the peptide ensembles, you tend to have more comprehensive coverage of the PepFlow conformations. Similarly, we found that on average, roughly 40% of ground truth conformations were captured when you sampled 1,000 conformations with PepFlow, and this value is continually increasing as you perform more sampling. As baselines, we compared PepFlow to molecular dynamic simulations. So we either ran molecular dynamic simulations for 100 nanoseconds in implicit solvent for the peptides in the test set, or 20 nanoseconds in explicit solvent for the peptides in the test set. And we found that the coverage by PepFlow was substantially higher at a fraction of the wall time. And these are all run, of course, on the same GPU to allow for reasonable comparison. Here's more extensive validation or comparison to the molecular dynamic simulation using different metrics. We see that the distribution of radius of gyration tends to be better with PepFlow. The KL divergence of the pairwise alpha distances tends to be better, and so does the coverage of both the ground truth and the PepFlow conformations when comparing the ensembles. Tends to be better with PepFlow compared to molecular dynamics. So how do we measure coverage is a question that's been asked in the chat. So it's basically just based on an armistice difference of 0.15 angstroms per residue, after you superimpose the conformations. So one final application we wanted to test PepFlow in was to assess whether or not. Yep.
Yeah, I have a question
Sure.
regarding that part. So when you measure the coverage, it's kind of a metric of the recall, right? Like how many of the actual conformation you can find. But is there, like, I don't know if maybe the KL divergence give you any information about that, but about the precision of like whether the conformation that you sampled are actually like the conformations of interest.
Yeah, so
like,
I think the coverage of PepFlow conformations is the precision, because it's how often the PepFlow conformations resemble something in the experimental ensemble, and the coverage of the ground truth conformations would be the recall. I don't know if that answers your question.
Yeah, that answers. And can you tell me like exactly why there are many points on this plot? Like, are they different peptides?
Yeah, so each of these points is a different peptide in the test data set.
Okay. So this means that like for some peptides you get like very high, like 75% both precision and recall, but for some other peptides you might get something different.
Yeah.
Do you know, like in general, if well, yeah, because here like you don't know how to correlate, like I don't know whether this peptide is that peptide, right? There's no way of saying like which one is which. Do you see an inverse correlation that like as you increase here, you decrease here, or like usually it's not really correlated?
So I haven't actually looked at that, but I do think you probably would see a correlation, simply because the predominant factor in determining the recall of the conformations is how extensively you or the number of experimental conformations you have. So necessarily you'd see for a higher number of experimental conformations, you'd probably see a lower recall, and you see higher precision. So there's almost certainly going to be a correlation between the two values. But it would be cool to look at that more in more depth.
Yeah, or at least like get an association between these two plots, like if you had, for example, like a triangle and another triangle, so we can match them, that'd be great.
Sure.
Yeah. Okay, thanks.
Right, no problem. Yeah, so one final application we looked at is to assess whether or not PepFlow could generalize to the sampling of macrocyclic conformations. So macrocyclization is a process by which covalent bonds are enzymatically added to non-adjacent regions of peptide, non-adjacent atoms and peptides, and from a therapeutic perspective or therapeutic development perspective, this confers a number of advantages, increases in vivo stability of peptides, their cell membrane permeability, and their oral bioavailability. So although PepFlow was trained on linear peptides, we reasoned that peptides, even linear peptides, should assume with some probability conformations that resemble their macrocyclized conformation. So based on this, we implemented an MCMC approach in latent space to try and identify conformations that satisfy certain bond length deviations. So we'd start with a random sample in latent space, assess the distance between the atoms that we want to be bonded, and make moves based on the metropolis criterion that it's defined based on the deviation between that distance and the desired bond length. And we also include an auxiliary clashes term to avoid movement to essentially garbage that contains a large number of clashes, or like a supercollapse conformation. So overall, we found that within a reasonable number of MCMC steps, we could consistently generate conformations that have the desired bond lengths. So the test set was divided into two different sets, things that are head to tail cyclized. So you'd have the N-terminal nitrogen covalently bound to the C-terminal carboxyl, and things that have side chain cyclization. So you'd have an atom in the side chain or covalently bonded to another atom in the peptide. So as a baseline for performance, we compare it to AlphaFold, which was recently shown to be able to model macrocyclic peptides really well by adding a positional offset to the positional encoding in the that's used when featurizing the input structure. And in general, we found that PepFlow doesn't perform as well as AlphaFold in terms of head to tail cyclization, often being outperformed by AlphaFold. But we do find that PepFlow, within 25 conformations, that's the number we generated for each test set peptide, PepFlow does consistently generate things that do look like the ground truth, suggesting that if we were to develop a better way to rank these conformations, we might be able to get improved performance. Since AlphaFold doesn't explicitly model sidechain atoms, we're unable to really use it to model sidechain cyclized peptides. So in this case, we simply compare it to linear peptides from AlphaFold, and we found that, as expected, PepFlow is much better able to generate reasonable peptide conformations. So that being said, in general, I hope what you got from this is that we developed a generalized all-atom Boltzmann generator to predict peptide conformations, and PepFlow is able to generate single structure predictions with a relatively high degree of accuracy, and is able to generate accurate conformational ensembles at a fraction of the running time of traditional approaches. So we're currently actively working to improve the framework. There's constantly improvements to both graph neural network architectures, as well as diffusion and flow frameworks that could be tried that could potentially improve the expressivity and performance of the model. Another big direction we're working on is trying to model bound peptide ensembles, because once we're able to do that, along with the unbound ensemble, we'll be able to perform thermodynamic calculations that could potentially be used to compute binding affinities and other metrics that would be useful in therapeutic development. And finally, we're also working on modeling peptides with different modifications, so non-natural amino acids, or in different conditions other than simply being solvated in water. So thank you for listening, and I'll be happy to take any other questions you have.
Very nice, very nice. I like this one a lot. Any questions from friends of the sun, or should we go into my high-level questions? Well, I wanted to ask, or I remember from the very beginning, I was enticed to ask, what do you think is still missing, or yeah, are the big things that we could do to maybe get like an AlphaFold level Boltzmann imitator for anything?
So I think, I don't know how viable the Boltzmann distributor framework is for larger proteins, because ultimately when doing the training by energy, you need to compute likelihoods, which is, extremely expensive. Even like, anytime you do, solve a dynamics function completely, it's going to significantly hinder training, especially when you get up to larger degrees of freedom. Here we're limited to 15 amino acid residues, and already feels like we're reaching the limit of what can reasonably be modeled computationally. So I think moving away from the Boltzmann generator does have the advantage of being very theoretically elegant. Like, you're directly trying to match the Boltzmann distribution, but I do think efforts like what was developed in the distributional graph former from Microsoft AI, which provides an alternative training by energy formulation that doesn't necessarily guarantee that you'll match the Boltzmann distribution, but still uses like physics inspired properties during training, is probably the direction that will be necessary. I mean, of course, if we have the generation of more ensemble data, either experimentally or computationally, would also greatly increase the ability to train these types of models. There is some ensemble data for proteins, but obviously the more the merrier when it comes to training these large networks.
Yeah, I would still call the score-based or score-based enhanced sampling thingy and the distributional graph former, I would still call those Boltzmann imitators. And
Yeah,
I would
I
also
think that's
not
fair.
call your model a Boltzmann generator, because you can't, for example, evaluate the likelihood of samples. Like, you can only evaluate the likelihood of samples that you generate, right?
Yeah. Well, here you can, yeah.
And
So
you
yeah, that's
also
the
don't have
that is the main difference ultimately.
you also don't have a likelihood that really a likelihood that you, or I assume that likelihood estimation isn't very accurate, and that you couldn't use it for reweighting your samples very well.
Yeah,
But
so that's like, given the fact that we use Hutchins estimators to compute the trace of the Jacobian, yeah, that significantly limits the utility of the likelihoods in reweighting samples or computing various statistics. But yeah, it does ultimately serve its purpose in allowing for training. But yeah, that is definitely a limitation of the current framework.
yeah, but
I guess
like
one other point, I do think, so there's also been a lot of success with just adding stochasticity to AlphaFold by subsampling MSAs, or by running the model with Traubelt layers to induce variability in the outputs. And those aren't like as well theoretically motivated, in that they have no like backing from biophysics, but they do seem to be working. So maybe it could be the case that the Boltzmann imitator route isn't necessarily the best in general.
Okay, well then let's maybe go with a last question from the chat. Perhaps a naive question, but can this type of hypernet producing parameters for diffusion networks be used to generalize across a range of physico-chemical conditions, like the pH, temperature?
Yeah, so I don't see why not. I think the biggest issue is with that type of training is you need, some
Data.
kind of, you need data to do the initial training by example from, a range of pH or temp conditions. I mean, you could always run molecular dynamic simulations, but that can be expensive. But if you do have the data, I don't see any reason why that wouldn't work. You might even be able to get away with doing training by example with one condition, and then having your force field be parameterized at different physicochemical conditions, and try and just do the training by energy. using those different conditions. I don't know how well that would work because all of the performance does come from the training by example phase, but that might be something worth trying.
So this has been pretty impressive to me. I mean, this has been like 40 pages of results or something. Osama really put together a large body of work here, I think. And I also like the general direction of all these Boltzmann imitators, as we've come to call them, I suppose. And yeah, if you know any interesting work like that yourself, then maybe head down to the description and find our Slack channels and our contacts and recommend the paper to us. And then we might very well invite the author and you can join the discussion with them as well. See you next week.